Hello everyone, welcome to our YouTube channel NEET YUG. I am Arun Kumar and in this particular video session I am going to explain the structure of Microsporangium. In our previous video session we have learnt the structure of anther. So this is the diagram of TS of young anther, that is the transverse section of young anther. Okay. And we have learnt that anther has two lobes. So anther is bilobed. So this is one lobe and this is the other lobe. Anther is bilobed and each lobe has two theca. So this is one theca and this is another theca. So total anther is dithecous. Correct? Total 4 theca will be there and in each lobe 2 theca will be there. Okay. And anther is tetrasporangiate. Very important, because in each theca there will be one microsporangium. So total 4 microsporangia will be there. So 1, 2, 3, 4. So what is the function of microsporangia? See, this is very important structure which actually is responsible for the production of microspores. Later the same microspore will produce or transform itself into pollen grains, that is the male gametophyte, which is very important for fertilization. Okay, first let us see the structure of it. You could see that I have used different colors here. See, the blue color one is actually the outermost layer of the microsporangium and it is called as the epidermis. Okay, so I'll just write 1 here. I'll write 1, the outermost layer, that is epidermis. So what is the function of epidermis here? First of all, the epidermis actually prevents the entry of water inside the microsporangiums. That is actually, it is water resistant. Okay, so it will not allow the water. So it prevents the water. So that is the important function. And another important function of this epidermis is actually it provides protection. So it is also a protective layer. So this is the thing you have to remember. So it is the protective layer. So I'll write protection of the microsporangium. Then we go to the second one. I have used the purple color here. This purple color structure is the second innermost layer, that is called as endothecium. Endothecium. And here endothecium particularly, what you have to remember is obviously it's going to protect the microsporangium. So it is the second layer of the microsporangium and it is fibrous in nature. Remember this, it is fibrous in nature. It is also going to protect the microsporangium. Okay, so that is the second. So I'll just write fibrous. And the third one, where you could see this particular structure is the third one, is the middle layer. Okay, so I'll write the middle layer. Normally epidermis and endothecium are single layered cells. This is single layered and this is also single layered, whereas the third layer, the middle layer, is mainly contains 2 to 3 layers of cells actually. So here actually you cannot see it, here only one line is there, but it is actually 2 to 3 layers of cells. Remember this. Particularly you may get one more question, what exactly this middle layer, what kind of cells are there actually? That is the parenchyma cells. The parenchyma cells actually makes the middle layer. The fourth one you could see, that is I use the pink color here. This is very important structure, that is the fourth one. So I use the fourth one is the tapetum. So fourth layer is tapetum. So we have learned that the microsporangium has 4 layers. The first 3 layers, 1, 2, 3, is mainly for protection. And it has got one more function, I'll tell you later. And the fourth one, the tapetum, has actually very important function. So we need a particular session to explain the structure as well as the function of this tapetum. Anyhow, I'm going to explain it a bit later. So, and here you can see that this is the pink color circle which actually surrounds a white color circle. Correct? You could see the white color circle here. Yes. So first we have to understand what is that white color circle. Okay, so this is a tissue called as sporogenous tissue. Sporogenous tissue. Okay, so the tapetum cells actually surrounds the sporogenous tissue, and what is there inside the sporogenous tissue? You could see many cells inside this tissue. All these cells are called as homogeneous cells or sporogenous cells, which is present inside the sporogenous tissue, and in future it's going to be a potential pollen mother cell. As the anther matures, obviously all these homogeneous cells actually becomes potential pollen mother cell. That means in future it's going to be a pollen grain, okay, or whatever the cells you see in future, it's going to be the pollen grain mother cell. Okay, I'll just write pollen grain mother cell. Okay, pollen grain mother cell, it is also called as microspore mother cell, remember it. That means actually there is some connection between the tapetum and this pollen grain mother cells, or maybe the sporogenous tissue. Very important function is it provides nutrition to the developing microspore. So it provides nutrition to the developing microspores actually. Other than that, it has 3 more functions. I'll talk about it later. Okay, so just remember. And anyhow, like let me just draw one more diagram. Okay, so this is now the microsporangium. Obviously I have explained all the 3 layers or the 4 layers actually. Okay, so you know what is this? Epidermis, and this is endothecium, and this is the middle layer, single layer, 2 to 3 layers, and the tapetum, it is also single layered. Okay, the first 3 layers I said, like the protection, not only protection, when the anthers mature to release the pollen grains, at that time the 3 layers actually dehydrate, and as a result helps in the dehiscence of the anther. Dehiscence. So what is the dehiscence? Dehiscence means the breakage. The anther should, the 3 layers should break. As a result, the mature pollen grains now are ready to be released out. Okay, so this is the very important thing to remember. So protection and dehiscence are the function of the first 3 layers, E, E, M. Whereas tapetum, obviously it's not going to protect the developing microspores, but it provides nutrition. How exactly it provides nutrition? This thing to remember here. Anyhow, I said like it provides nutrition. So let me just write here, it provides nutrition by absorbing some nutrition from the middle layers. So middle layers are the one which provides some nutrition to the tapetal cells, and the tapetum now provides nutrition to the developing pollen grain. And one more thing to remember is, please look into this structure, and it exactly looks circular. So microsporangium is circular in outline. I have a model to explain the concept. See this, I have prepared this model. Okay, so this is one microsporangium, and you could see the red color blocks are the epidermis. How many layers are there? Only one. And you could see the purple color blocks are actually the endothecium, and the third layer, the yellow color blocks, are the middle layers. How many layers are there? 1 and 2. That means 2 to 3 layers. And this one, the black color one, you see here is, that is actually the tapetum. And the next thing what you could see here, the pink color circle, that is the sporogenous tissue. This is the site where the microsporogenesis takes place, that is microspore formation takes place, and the same microspore in future becomes the pollen grains. These tapetal cells actually provide nutrition to the developing microspore mother cells, or microspores particularly. How? Because they absorb the nutrients from this particular layer, the middle layers. The function of these 3 layers, 1, 2, 3 layers, for the first 3 layers, protection and dehiscence of the anther, the tapetum, nutrition to the developing microspores or pollen grains. Remember this. Now let us see the structure, types and functions of tapetum. Very, very important actually. That's what actually I have erased all these things because I need space. And you could see here I have written structure. So let us see the structure of tapetum cells. Okay, normally I said like tapetum cells actually surrounds the sporogenous tissue, which is single layered, single cells. So let me just draw one tapetum cell. So normally the tapetum cells are big, they are large cells actually. That means you may get this question, which is the largest cell in microsporangium? Tapetum is the largest cell in microsporangium. And one more thing I want to tell you that it has got more than one nuclei. Okay, so it could be multinucleate or it could be binucleate, normally multinucleated. So I'll just write here, it is multinucleated. Okay, so it has got a dense cytoplasm, more cytoplasm is there. So it has got more cytoplasm because it has to provide the nutrition to the developing pollen grains, correct? If you take this tapetum particularly, the nearest cells are the sporogenous cells, correct? The sporogenous tissue. So that's what actually they have to provide the nutrition, and hence they have got dense cytoplasm. One thing I want to tell you here is, the tapetum actually exhibit polyploidy condition. This is very, very important, and also it exhibit polyteny condition. Okay, so what is this polyploidy? For example, say in our cell we have a total of 46 chromosomes. Each cell has 46 chromosomes. That means our ploidy is diploid, that is 23 plus 23, correct? N plus n, that is 2n. So that is diploid condition. If I have 69 cells in 69 chromosomes in one cell, so that is 3n. Or if I have 92 chromosomes in one cell, that is actually 4n. So if I have like that condition, then it is called as the polyploidy. So that means here you could see it is binucleated, it is multinucleated, and obviously the chromosome sets will be more actually, hence it is polyploidy. Remember it has got extra set of chromosomes. Okay, so I'll just write here extra set of chromosomes, that is polyploidy. And then what is this polyteny? Here polyteny means obviously inside the nucleus, and obviously the chromosomes will be there, correct? So chromosomes will be there, and obviously the chromosomes means nothing but DNA. So this DNA obviously undergoes replication. As a result of replication, the chromatid material grows, and then once they grow, then obviously they have to detach and form a new chromosome. But in this case they don't form the new chromosome. Instead of that, they remain attached to this chromosome. As a result, the chromosome looks huge and large. So this condition is called as the polyteny. So polyploidy, polyteny are the important characters exhibited by only tapetum cells in Microsporangium. Okay, so what is the reason behind this polyteny? Simple, that is called as endomitosis. Remember this. So which cells in Microsporangium exhibit endomitosis? Tapetum. Endomitosis means it is not cell division, it is actually a kind of replication of the DNA, replication of the chromosome, that is called as endomitosis. Endomitosis. And here they don't separate. As a result, polyteny condition will be seen in the tapetum. And the types, okay, so here there are two types actually. So I'll just write there are two types. The first type is called as glandular. It is also called as secretory, glandular tapetum, and the second type is actually called as amoeboid, or it is also called as plasmodial tapetum. Amoeboid or plasmodial tapetum. Okay, so glandular tapetum, okay, fine, we'll draw the one tapetum here. So this is the tapetum having dense cytoplasm. Okay, so it is going to release a type of body called as Ubisc bodies. What kind of bodies? Ubisc bodies. How they are normally these cells actually secrete Ubisc bodies, where here when they secrete the Ubisc bodies, the cell wall doesn't break. So here what happened, the Ubisc bodies actually they come out of the cytoplasm and particularly the plasma membrane and the cell wall, and finally it is released out. So you know that the nearest cells are microspore mother cells, that is porogenous tissue. So here the Ubisc body is actually, for example, I'll just use this space here. So let us assume that this is the sporogenous tissue. Here actually you could see potential pollen mother cells are there. Okay, let us assume that now they have entered into microsporogenesis, where microspores are being produced from pollen mother cell at that moment. Okay, so these are all, let us say, glandular tapetum. Okay, so now they are going to produce the Ubisc bodies. Here the cells will not break. Instead of that, only the Ubisc bodies will be released. I need a chalk piece here. Okay, so here what happens actually, the pink color ones are the Ubisc bodies which will be released, and these Ubisc bodies actually surrounds each microspores like this. And what is the function of these Ubisc bodies? Oh, sorry, I forgot to tell you that exactly it is called as pro-Ubisc bodies. Once they actually surrounds each microspores, then we call it as Ubisc bodies. Okay, so released ones are pro. Then after that, matured ones are actually Ubisc bodies. Okay, fine. Here actually, once they surrounds all microspores, obviously they will form sporopollenin of the pollen grain. Sporopollenin is actually the outermost layer. Exine is there, right? In the exine there is one organic material is there, that is sporopollenin. Who makes sporopollenin? These hibiscus bodies released from the tapetum. Okay, another one, the amoeboid. Here again I'm going to draw this. Here what happens, actually the same mechanism, but here the cell wall breaks. As a result, the entire protoplasm, okay, so this is the protoplasm, entire protoplasm released out. So what is the shape of the protoplasm now? Because of the rupture of the cell wall, it looks like amoeba, right? So hence it is called as amoeboid. Plasmodial, entire protoplasm is released out, and obviously the tapetum cells actually dies, and now all will form the same mechanism. Actually it surrounds the developing microspores and form the sporopollenin. That's actually the thing. The function is same, but the mechanism of release of the hibiscus bodies is entirely different. That's it, okay, in both the types. And this one is the common one, very common. It is normally seen in all the angiosperms. This one normally seen in the primitive angiosperms. Normally you don't see them now. It is normally seen in the primitive cases of angiosperms. Okay, now the functions. There are 4 functions, remember it. One function I have already told you. The first function is provide nutrition to the developing pollen grain. How? From the middle layers. They extract or absorb the nutrients from the middle layer and they supply nutrition to the developing pollen grains. The main function is to provide nutrition to the pollen grain, developing pollen grain. Okay, and when they absorb the nutrition from the middle layer, what will happen to the middle layer? Obviously middle layers are made up of parenchyma cells. They degenerate. So that means in the mature anther you don't see the middle layer. You will see epidermis, you will see endothecium and tapetum will be seen, but middle layer will not be seen because they get degenerated, because obviously this particular tapetum absorbs all the nutrition from the middle layer and they get degenerated. That's it. The second function is they actually form the pollen kit. Where exactly you see the pollen kit? Pollen kit means actually let me just write. Okay, so here the pollen grains will be produced once the anther matures after the process called as microsporogenesis. And these pollen grains, for example, if the plant is entomophilous, if the plant exhibit entomophilous flower, what is the meaning of entomophilous? The flower which is dependent on insects for pollination. Okay, that means here the insect should come and visit a flower, and then actually they have to take some pollen grains. If they take 1 or 2 pollen grains, it's not useful for the flower. That means they have, when they visit, obviously they have to take more number of pollen grains. So very simple, what happens actually when these pollen grains are produced, that particularly the tapetum cells actually produces a lipid plus carotenoid rich substance which actually surrounds a cluster of pollen grains, which is normally sticky. This is called as the pollen kit. It is also called as pollen coat. Remember this, it is also called as pollen coat. Pollen kit, it is also called as pollen coat. That is the function of the tapetum. The third function is they secrete hibiscus bodies. What is the function of hibiscus bodies? I have already told you when I explained the types of the tapetum. Hibiscus body is actually helpful in the formation of sporopollenin, one of the toughest organic material present in the exine of the pollen grain. That means when the microspore mother cell is undergoing microsporogenesis to produce microspores, at the time the sporopollenin formation takes place, which surrounds each microspores, and later when the microspores differentiate to produce pollen grains, at that time the sporopollenin becomes the exine. So it will merge with the exine and makes the exine the toughest, unbreakable. Even any kind of enzyme cannot break it. So because of this sporopollenin secreted by the hibiscus bodies, obviously released from the tapetum cells. Okay, remember it. The fourth one is it releases one enzyme called as scalase enzyme. Remember this, scalase enzyme is secreted by the tapetum cells. And why we need this scalase? Because when actually the microsporogenesis takes place, at the end of the microsporogenesis, and you see from one microspore mother cell 4 microspores will be produced. So this is a pollen mother cell. It is also called as microspore mother cell, and as a result of meiosis, that is microsporogenesis process, and what you see is actually a microspore tetrad is produced. I'm going to explain in detail later, fine. So at the time each microspores are actually attached to another microspore, so it cannot, it's very difficult to detach. So why? Because there is a polysaccharide bond between each microspore. What is that polysaccharide bond? That is called as the callus. Okay, so we need to break this callus. I have to remove this callus. How we can remove this callus? Very simple. That is callase enzyme. The callase enzyme will degenerate the callus, and as a result these microspores now get separated. That's it. It's like, you are removing the callus by the callase enzyme. That is actually the function of the tapetum. Very important. So please note this point, take a pen and paper and write everything. Very, very important for your NEET.